For the first time, a research team from China used a new technique to fix a blood disorder in human embryos. The scientists performed chemical surgery, a procedure that rewrites errors in genetic code instead of snapping and replacing strands of faulty DNA, which is the central strategy employed by the CRISPR gene editing system. The early results look promising, but this technology is still a long way from prime time. Brazilian Jiu-Jitsu is a fascinating and even unconventional martial art in that its practitioners, rather than coming out all guns a-blazing with fists, knees, and elbows, use their opponent's weight and strength against them. This new gene editing technique, which was introduced last year by a Harvard research team led by David Liu, is a lot like Jiu-Jitsu in terms of its subtler approach, where CRISPR slashes sloppily through DNA like an unsharpened knife. The chemical surgery technique, as Liu calls it, actually goes into the code of DNA itself, flipping misspelled letters or bases into their correct form. A team of researchers from Sun Yat-sen University in China are now the first to use Liu's invention on an actual human embryo, though the modified embryo was not implanted into a surrogate. As described in the science journal Protein and Cell, the technique was used to correct beta thalassemia, an inherited blood disorder that affects 1 in 100,000 births. This is the same team, by the way, that was the first to use CRISPR on human embryos, though that pioneering attempt to fix abnormal beta thalassemia genes was met with limited success. By turning to chemical surgery, a technique otherwise known as base editing, the researchers were hoping to get superior results. The new technique is not meant to replace CRISPR, but it could be used to fix inherited disorders caused by single mutations, and in a way that produces fewer unpredictable results. The scientists are claiming limited success, but because the technique sometimes caused new mutations instead of fixing them, proclamations that this is the next big thing in gene editing should be met with trepidation.